This is the biggest learning objective here is actually talking about what is occurring in photosynthesis. What is the process that is actually happening? And there are two stages, and I'm going to tell you about them and where they occur in different parts of the chloroplast. So photosynthesis has two stages, kind of illustrated here in this cartoon. There are the light reactions, also called the the light-dependent reactions. This is the photo part. This is where light actually is absorbed by the chloroplast in the chlorophyll. And then there's the light-independent reactions called the Calvin cycle, and this is the synthesis part, or where we actually use that energy harvested by the light reactions to make the sugars that the plant uses for energy, that we use the plant surplus for our own energy. So we're going to talk about two stages, kind of in order: the light reactions and then the Calvin cycle. Okay, so one of the points of this objective, or this learning objective, is for you to understand what is going into and coming out of the photosynthetic process. And the light reactions, water comes in and oxygen comes out. Oxygen is a byproduct of photosynthesis. As it turns out, oxygen is not necessary for photosynthesis. It's kind of a waste product, which for us happens to be very useful, as we saw when we talked about cellular respiration. Oxygen makes it so that we can produce lots and lots and lots of ATP. Light is also one of the parts of the light reactions, which makes sense because they are the light reactions. And I'll tell you how light gets absorbed, that energy absorbed, and that happens in the thylakoids. The Calvin cycle occurs out in the stroma, away from the thylakoids, using the products of the light reaction. And the main products of the light reaction are ATP and this molecule called NADPH, which should sound kind of familiar because we saw something just like it in cellular respiration, only without that P there. In the Calvin cycle, carbon dioxide is brought in through the stomata as a gas, and sugar is the product of the Calvin cycle. So we can store up that energy through photosynthesis as sugar that can then go into cellular respiration. So what you see in this picture is a diagram of chlorophyll, which is that main photopigment that plants use for capturing the energy of photons. So chlorophyll A and chlorophyll B are mostly the same molecule. The only difference is in this little part up here, you've got a CH3 in chlorophyll A and a hydroxyl group in chlorophyll B. But there's this ring of nitrogens that have a magnesium atom in the center. And that magnesium atom is the thing that gets excited. The electrons in that magnesium atom get excited by photons of light. And then you've got this long hydrocarbon tail, which helps to anchor the chlorophyll molecules in the membranes of the thylakoids. Remember, membranes have that hydrophobic part, and just like we could anchor enzymes in the membrane by using hydrophobic amino acids, using this hydrophobic tail, the chlorophyll gets anchored within the thylakoid membranes. So here are a couple of illustrations of how chlorophyll receives light. So we've got this set of molecules embedded in the thylakoid membranes called a photosystem. Each of these green dots represents a molecule of chlorophyll. The photon comes in and excites the electrons around that magnesium atom, and that state of excitement can be bounced around from one chlorophyll molecule to the next until it gets to the center of the photosystem. And those very high energy electrons can then be transferred to this thing called a primary electron acceptor. And this primary electron acceptor is very, very powerful, very, very powerful oxidizing agent. And I'll tell you how powerful in a second. But over here, this is a ribbon model showing the different proteins and chlorophyll molecules that are all bound up in that photosystem. Okay, so there are two types of photosystems, photosystem 2 and photosystem 1. Photosystem 1 was discovered first, even though it comes second in this whole process. So photosystem 2 receives the light first, and that photon of light excites the electrons in chlorophyll, and they bounce that state of excitement around, bouncing electrons around, until they get to the center of the photosystem. And those electrons are transferred to a primary acceptor, which, remember, I said that's a strong oxidizing agent. How strong is it? It's so strong that it can oxidize oxygen. Remember, oxygen is very greedy for electrons, but the primary acceptor can actually pull electrons away from oxygen in water. In water, the oxygen is relatively reduced because it's holding on to those electrons more than the hydrogen atoms. But in oxygen gas, they share those electrons evenly. So in oxygen gas, it is a relatively oxidized form for those electrons in the oxygen gas. So that's where the oxygen we breathe comes from. It happens in this very first step of photosynthesis as light bounces the electrons around, and it can pull the electrons off of the oxygen in water, releases oxygen gas, and then those electrons can be passed along an electron transport chain, just like we saw in cellular respiration, and producing ATP in much the same way. So passing the electrons along a series of multi-protein complexes, not the same as the ones that are in respiration, but similar to them. And then we get to photosystem 1. And those electrons still have a lot of energy, even though they have done the work of, just like we saw in cellular respiration. But a second photon of light can re-energize those electrons, and then they can be put onto another electron transport chain that will transfer those electrons onto a molecule called NADP+. And NADP+ is an electron shuttler, just like NAD and NAD+ and FAD are electron shuttlers. Only there's a P there because they have an extra phosphate group. But you can remember that P stands for plant. And I'm going to talk more about NADPH in a minute or two. So, but the NADPH is absolutely essential for going into the Calvin cycle, as is the ATP. So here's what it looks like in another type of representation. You can see we've got our two photosystems. We're looking at the coin-shaped thylakoid, which has a space on the inside, and then the outside is in the stroma, maximizing surface area. And just like in cellular respiration, we are putting those electrons to work by pumping protons from one side of a membrane to another to generate a concentration gradient. And That concentration gradient drives a molecule of ATP synthase, just like we saw in cellular respiration in producing ATP, only we call this photophosphorylation instead of oxidative phosphorylation. We can recycle those electrons that did the work to pump the protons into the thylakoid space. So it's got a high proton concentration, a low pH, it's strongly acidic, but those protons can get out through ATP synthase. And that produces ATP, and all of that goes to the Calvin cycle. So you're probably thinking that there are a lot of similarities between cellular respiration and photosynthesis, and you would be absolutely right. You can see here in this cartoon sort of comparing the structure of the mitochondrion and the chloroplast. And you can see in both cases, you've got a pH gradient that is being used to drive ATP synthase. The source of energy is a little bit different, very different actually, between the two. But a lot of the other moving parts are very similar. And that we're using a membrane to maintain a gradient so that we can spin ATP synthase to produce ATP. So here is chloroplast ATP synthase. We've got the protons that are stuck inside the thylakoid lumen that will move through ATP synthase to try and get into the stroma to try and move towards equilibrium. But as they do that, ADP and phosphate molecules are going under these knobs here and just producing a lot of ATP. And that ATP is then going to go into the second part of the process, which is the Calvin cycle. NADP plus is the fancy bus. NADP plus, like NAD plus, accepts electrons, but NADPH carries electrons that have a lot more potential energy than the electrons that go on NADH. Now, this should make sense to you because NADPH is being used to produce the sugar that we then use for energy in cellular respiration. And the laws of thermodynamics say that when you transfer energy, you lose some. No energy transfer is 100% efficient. So burning glucose to release energy can't release as much energy as the process that went into producing it, because producing glucose is increasing, is ordering the universe. And the energy for that comes from sunlight. So anyway, another way of saying that is that NADPH carries much higher energy electrons than NADH or FADH2. Okay, so that reducing power, those high power electrons on that fancy bus are then going to go to the Calvin cycle, where with ATP, they're going to produce some sugar. Okay, now on to the Calvin cycle. Oh, this is a lot of text on one slide, so we'll take it slow. The Calvin cycle, again, that's the synthesis part of photosynthesis. This is where we take all that energy that we harvested and we use it to make sugar. Now, the sugar that we produce is not glucose directly. It's another type of sugar called glyceraldehyde 3-phosphate. It's a 3-carbon sugar, but it's really easy to turn that into glucose for storage purposes. The carbon that is used, it comes from carbon dioxide. It comes from the atmosphere through the stomata, but it's a very, very highly oxidized form of carbon. So we need to reduce the carbon in carbon dioxide. We'll use those powerful electrons on the fancy bus to do that. It's also going to take a lot of energy in the form of ATP. It's also going to require an enzyme that's called Rubisco. Rubisco is an abbreviation for a much longer name that I'll tell you in a little bit. But that is a very important enzyme that makes all of this happen. There are 3 phases that are very important for us in the Calvin cycle. The first part is carbon fixation, or bringing the carbon into from an organic molecule. Then we take that carbon and we reduce it, so we add electrons to it. And then we have to get back to the molecule that is going to accept new carbon atoms, new carbon dioxide molecules. And that's the regeneration of ribulose bisphosphate, which is the carbon dioxide acceptor. Okay, so first phase in the Calvin cycle is carbon fixation. So we start off with a 5-carbon molecule called ribulose bisphosphate, abbreviated here as RUBP. You can see it's got 2 phosphate groups on the ends of that 5-carbon chain. But in order to make this work, we need 3 molecules of Rubisco, and we need 3 carbon dioxide molecules. And the reason that we need the math to work out that way is because when Rubisco puts a carbon dioxide molecule onto RUBP, it makes a very unstable product that pretty much spontaneously breaks down to form 6 molecules of a 3-carbon molecule called 3-phosphoglycerate. So instead of having 6-carbon molecules, these break down into 6 3-carbon molecules. Okay, so the next step is we need to phosphorylate 3-phosphoglycerate to give it a phosphate at both ends. Now this molecule has a lot of free energy attached to it, but we need to reduce the carbon on it. So the next phase is using those very fancy electrons from the fancy electron bus to reduce that carbon to make a molecule called G3P. And we have 6 molecules of G3P. And one of those G3P molecules can then leave the Calvin cycle. All right, so to make it a cycle, we still have to finish what's over here in this section over here. So we have to regenerate ribulose bisphosphate. The way that we do that is those 5 remaining G3P molecules, we're going to kind of shuffle them around again. We're going to use the ATP energy to phosphorylate them. We're going to take these 5 3-carbon molecules, shuffle them around to make 3 5-carbon molecules. So there's a lot of shuffling around of carbon skeletons that makes this happen. So there's a lot of, you get the 5 by 3 to make 3 times a 5-carbon molecule, and you have to take those 3 6-carbon molecules to make 6 3-carbon molecules, and you can see why this is kind of a complex process. And the experimental design that went into figuring all this out was truly noteworthy, truly amazing, landmark watershed event for science. Okay, so Rubisco is this protein, this enzyme that I was telling you about. It stands for ribulose bisphosphate carboxylase oxygenase. And that's a mouthful. But ribulose bisphosphate is that 5-carbon sugar that we add CO2, carbon dioxide, to. So that's the ribulose bisphosphate carboxylase. And I'm not going to go into the oxygenase part of it, although it's another interesting story. Rubisco is thought to be the most abundant enzyme on Earth. It does a job that is so fundamentally important that there has to be a lot of it, and it's also not a very efficient enzyme. So chloroplasts have to make a lot of copies of this particular enzyme. So that's why it's the most abundant on Earth. And what it does is it fixes carbon dioxide, attaches it to this 5-carbon sugar to make new sugar. Okay, so that was the process of photosynthesis. For our next learning objective, I'm going to show you a couple of other short YouTube clips that explain things I think better than I would be capable of doing it. And then I will come back and kind of finish off with a few points about how photosynthesis counteracts the greenhouse effect.